Hi everyone, my name is Vineet Agrawal. Thank you for joining us on this tour of the Pulmonary Vascular Biology Lab at Vanderbilt University. The lab is run by Hemnes and James West, so come on in and let's take a look. All right. Well, welcome to our Pulmonary Vascular Biology Lab. As you can see, our lab consists of one large room with multiple bays. At the end of each bay are multiple desks at which people sit. And so you can see our group consists of a very diverse and large group of people who work together to improve our understanding of pulmonary vascular biology. Let's come on in and take a look at a bay. All right, well, this is one of the many bays in our Pulmonary Vascular Biology Lab. The bays are where we do all of our wet lab work. What that means is we run reactions with cells, tissue, proteins, RNA, and DNA. Because these particles are so small, we can generally run them in very, very small tubes. Okay. Well, let me show you some of the tools that we have in our laboratory. But before we get started, safety comes first. One of the most common tools that any biology lab will have is what we call a set of pipettors. What these are tools that allow us to very, very finely transfer certain volumes of fluid. For example, if we wanted to transfer 0.5 milliliter fluid from one tube to another, what we would do is pick it up with our pipetter, and you can see it's in our pipette tip, and carefully pipette it into another container. Oftentimes, when we're setting up our reactions, we have to mix multiple liquids or products together and have to ensure that they're thoroughly and well mixed. The way that we do that is we use various mixers that we have in our laboratory. And this is one example of a mixer where once you push down, fluid will start to mix within a tube, leading to a uniform mixture. Other ways that we can mix samples, or if we need to bring samples to the bottom of a tube so we can remove them, is through a centrifuge. We have various types of centrifuges in the lab, anywhere from large to small, but I wanted to show you the smallest one that we use here. What we can do is we can take tubes, put them in, shut the lid, and what it'll do is it'll spin everything down to the bottom of the tube. Well, welcome to our tissue culture room. This is the room where we work with growing and maintaining and studying live cells. The way that we do that is that we have to maintain sterile conditions when we work with these cells, and we do so in one of these devices that we call a laminar hood. So in this device, whenever we're working with cells, we work with them with our hands underneath the glass and airflow to prevent any sort of contamination from coming within the hood. The cells are all worked with inside on the metal surface, as you can see. And when we're not working with the cells, we make sure that these hoods stay sterile and decontaminate them with the UV light, which is why you see that the inside of the hood appears blue right now. Well, when we're not working with the cells in the hood, where do they live? Well, come on over and let's take a look at one of the incubators where we keep our cells. So as you can see from the inside of the incubator, there are multiple shelves on which the various cells live. They live within either flasks or plates on each of the shelves, and they live on a monolayer, kind of a single layer at the bottom of the plate or the flask, with media or some sort of liquid to give them nutrients on top of them. In order to maintain the optimal conditions for growth and survival, each of these incubators has a certain temperature that they're set to, a certain amount of humidity, a certain oxygen concentration, and a certain carbon dioxide concentration that are closely monitored and closely set by the incubator. Well, I told you that the cells, the DNA, the RNA, and the protein that we work with are very, very small. So small that the naked eye can't see them. So how do we see them when we want to work with them? Well, we do so particularly with live cells using a microscope. So here we have a flask that has cells on the bottom and media on top. So how do we look at them? Well, what we do is we put them on the plate here on the microscope, and using light that shines through the microscope, we can look through the binoculars and see the cells underneath. So, when we're working with DNA and RNA, we have to have a way to measure the amount of it. We do that using this small machine here that allows us to measure very, very small amounts of DNA and RNA. And then when we start to study them in a reaction, we put them into a machine that we call a polymerase chain reaction machine, otherwise known as a PCR machine. What this does is, with the right reagents in each tube, allows the DNA and the RNA to replicate and grow. And we use a special dye to monitor that process. And we can, as you can see here, we can then watch as each of the tubes has increasing amounts of DNA and RNA that go into each tube. Well, this was the tour of our Pulmonary Vascular Biology Lab. We really hope you enjoyed it. Thank you for taking the time to join us.
Welcome to the Vanderpool Lab Virtual Tour. I'm Rebecca Vanderpool. I'm an assistant professor at the University of Arizona in the Division of Translational and Regenerative Medicine, as well as biomedical engineering. My lab focuses on investigating right-sided heart failure in patients with pulmonary hypertension. We're located in Tucson, Arizona, where it's sunny over 350 days of the year. We're at the University of Arizona, where we have some great expanses, and my lab is located on the University of Arizona Health Sciences campus. The research goals in the lab are to integrate imaging and hemodynamic data to quantify right ventricular function in pulmonary vascular disease, to investigate mechanisms of right ventricular dysfunction, and to identify novel therapeutic targets in the right ventricle. We do this by quantifying MRI imaging to assess ventricular dynamics. We use structure data to assess changes in the pulmonary circulation, and we use pressure and volume data to assess right ventricular function, both systolic and diastolic function. We investigate these changes in the development of right ventricular dysfunction. In the lab, we use tools such as physiological modeling, exercise hemodynamics, cardiovascular hemodynamics, right ventricular function, to assess the effect of pulmonary hypertension in both the clinical setting as well as animal models of pulmonary hypertension. We also use cardiac imaging in the assessments. A great way to get a feel for what we do and how we do it is to take a virtual tour of the campus. We will start out in downtown Tucson and walk towards campus. So this is going down 4th Avenue, where there's a lot of restaurants, businesses, tattoo parlors, and we turn the corner to University Avenue. There's a lot of frat houses, some coffee shops, and other stores going to the University of Arizona campus. In my lab, we focus on cardiopulmonary interactions. And to do this, in our various lab spaces with scheduling and everything else, we use animal models of pulmonary hypertension, isolating out the lungs and heart to assess pulmonary vascular structure, We look at MRI imaging to quantify also the vascular structure. And then we present it at posters and poster sessions around campus and internationally. We'll assess changes in blood samples, as well as write papers and grants to further our research. One great thing in Arizona are their sunsets, where you get the beautiful southwest colors that come through, as well as the cactus. Now we're leaving the campus. And thank you for your attention with this virtual tour of my lab at the University of Arizona. Of course, all of this research isn't possible without my lab members, collaborators, as well as funding sources. Thank you for your attention.